Ever stop to think about the incredible, invisible story written within every living thing? Not in books, but, in the very code of life, are genes, and the genes of everything else on this planet.
Yeah, it's pretty amazing stuff.
You've landed on the deep dive because, like us, you're hungry to understand the world and eager to get to the heart of complex topics quickly and effectively.
And that's exactly what we're doing today. We're diving deep into molecular phylogeny and the forces of molecular evolution. Think of this as your express route to grasping how we can trace the relatedness of all life by examining our fundamental building blocks, our molecules.
Right.
We've been exploring a really insightful lecture series on this, and there are some truly game-changing ideas in there.
Absolutely. So molecular phylogeny might sound a bit sci-fi, but it's really just about using things like DNA and protein sequences to figure out how different organisms are related.
Exactly.
It's the modern way to draw the ultimate family tree of life. Instead of just looking at bones or feathers, what
Sure.
scientists call morphology, we're looking at the actual instruction manuals.
And this molecular approach has allowed us to answer some truly astonishing questions. For instance, you can trace the lineage of almost any gene, maybe even one you're particularly interested in, and see who else carries a similar version and how it's changed over, well, vast stretches of time.
It gets even more surprising. By looking at these molecular connections, scientists have discovered some really unexpected family ties in the animal kingdom. Would you ever guess that whales, dolphins, and porpoises are actually close cousins of cows?
I know, right? They look so different on the surface.
Totally different.
Exactly. And it's not just about these big, surprising relationships. We can also investigate the origins of things that directly affect us, like viruses. Molecular analysis has been absolutely critical in understanding where HIV came from and how it has evolved.
Plus, if you zoom way out, molecular phylogeny helps us piece together the entire history of life on Earth, from the very first spark to the amazing variety of creatures we see today. It even helps us understand the relationships of animals that are now extinct, like the quagga, that fascinating creature that was stripy like a zebra at the front but looked more like a horse towards the back.
Ah, yes, the quagga. Our source even had a picture, a reminder of these lost branches on the tree of life.
Right.
And it was molecular data that really nailed down its close connection to zebras and horses.
Wow.
And what's really empowering is that you can even explore this yourself. If you're feeling curious, there's software out there like Mega, that's MEGA. You can find it at megasoftware.net that lets you play with this kind of data and even build your own evolutionary trees.
Build your own trees. That's cool.
Yeah. Our sources had examples of these trees specifically for genes called OX1R and OX2R. You could see how different creatures, from us and our furry friends like dogs and cows to birds, fish, frogs, and even insects, are positioned based on how similar or different these genes are.
So the tree structure shows the relationships.
Exactly. The length of the branches tells you how much evolutionary distance there is, and the scale bar, like 0.010 substitutions per site, gives you an idea of the amount of change. And those numbers at the forks in the tree,
Yeah, what are those?
those tell you how confident scientists are about those particular relationships. It's called bootstrap support.
Got it. Okay, so we're using these molecular clues to reconstruct the great family tree of life. But what actually drives these changes in our molecules over time? What are the engines of evolution at this fundamental level?
That's the heart of molecular evolution, and our sources point to three main players: mutations, selection, and genetic drift.
Mutation, selection, drift. Okay.
It's crucial to remember that these forces don't act alone. They're constantly interacting, sometimes pushing in the same direction, sometimes opposing each other, creating a kind of balance in the frequencies of different versions of genes, what we call alleles.
A dynamic balance.
Precisely. To get a baseline, it's useful to think about something called Hardy-Weinberg equilibrium, which describes a theoretical scenario where in a perfectly stable population without these evolutionary forces, gene frequencies would just stay put generation after generation.
Like a starting point.
Exactly. But in the real world, these forces are always at work, nudging things in different directions.
Okay, let's unpack one of these forces. Genetic drift. It sounds a bit like things just randomly changing.
That's a great intuitive way to think about it. Genetic drift is essentially random fluctuations in how common different versions of a gene become in a population, and it happens purely by chance during reproduction. It's a stochastic force.
Stochastic, meaning random.
Yeah, driven by randomness, not by whether a particular gene is better or more advantageous.
So it's not about survival of the fittest at this point.
Not directly, no. The mechanisms behind drift are all about luck, really. Think about who happens to mate with whom just by chance. Some individuals might leave behind more offspring than others. Then there's fertilization itself. Even with the normal shuffling of genes, the exact combination an offspring gets is a matter of chance. Mendelian segregation. And even something as simple as some families having slightly more kids than others, purely by random variation, can play a role in the next generation's gene pool.
Okay, so even if you have a perfectly good gene, it might just become less common in the population simply due to a string of, well, random events.
Exactly. And the power of this randomness is really tied to population size.
Ah, right.
In large populations, these random ups and downs in gene frequency tend to be small and kind of average out over time. But in small populations, a chance event can have a really big impact, even leading to some versions of a gene disappearing
Yeah,
altogether.
just by chance.
Yeah. The smaller the population, the more influential drift becomes. Think of it like flipping a coin a few times versus flipping it 1,000 times. You're much more likely to get closer to a 50-50 split with more flips.
That makes sense. A random event is going to be a much bigger deal when there are fewer individuals around. Now, you mentioned natural selection as another key force.
Yeah.
How does that contrast with this random drift?
That's a fundamental difference. Our source material had a really helpful way of comparing them. The core distinction is the process itself. Genetic drift is random,
Right.
while natural selection is non-random.
Non-random, okay.
The cause of drift isn't anything to do with the environment, it's just chance. Natural selection, on the other hand, is directly driven by environmental pressures. The fact that some individuals have traits that make them better able to survive and reproduce in their particular surroundings.
So drift is like a car without a steering wheel, just bouncing around, whereas selection is like a car being driven in a specific direction by the environment.
That's a great analogy. Drift is non-directional
Yeah.
and therefore doesn't inherently lead to adaptations traits that improve survival.
Right.
Natural selection, however, is the engine of adaptation, gradually shaping organisms to be better suited to their environment. And the fate of genes is very different under these two forces. With drift, even beneficial genes can be lost from a population just by bad luck.
Wow, even good ones.
Yeah. With natural selection, it's survival of the fittest. Genes that provide an advantage are more likely to be passed on. Finally, think about genetic diversity. Drift tends to reduce genetic diversity within a population over time as some genes become fixed and others disappear randomly.
Less variety.
Right. Natural selection can initially increase diversity by favoring different traits in different individuals, but then refines it over time, favoring the most advantageous versions.
Okay, so drift is the unpredictable element, and selection is the guiding hand of the environment. Now, how do these two interact? You mentioned they can create a dynamic equilibrium.
One important concept illustrating this interplay is mutation-selection balance.
Okay.
Mutations are constantly introducing new versions of genes into a population, and some of these new versions can be harmful. Selection acts to weed out these harmful versions.
Great.
Mutation-selection balance is the point where the rate at which new harmful genes are created by mutation equals the rate at which they are eliminated by selection. It's like a leaky bucket where mutations are adding water and selection is slowly draining it. You can reach a stable water level.
So even harmful traits can persist at low levels in a population.
Exactly. A constant reminder of the trade-offs inherent in the evolutionary process.
So there's always a low level of these not-so-great genes hanging around because they keep popping up through mutation.
Precisely. Our source gives an example using some simple math. Let's say U represents the rate at which a new harmful version of a recessive gene appears through mutation.
Okay, U for mutation rate.
And let's say IS is a measure of how much selection works against individuals who have 2 copies of this harmful gene, since it's recessive.
Is for selection pressure.
Right. At equilibrium, the rate of new mutations equals the rate of elimination by selection, which can be expressed as US score, where Q is the frequency of individuals with 2 copies of the harmful gene.
U equals SQ squared.
Yeah. This allows us to calculate the equilibrium frequency of that harmful gene, Q, as the square root of S.
Q equals square root of U over S.
And in a really extreme case, if having 2 copies of the recessive gene is lethal, meaning S equals 1, then the frequency of that gene in the population, Q, simply becomes the square root of the mutation rate, U.
Just the square root of the mutation rate. Wow, that's a powerful little equation showing how these opposing forces can balance each other out. What about the interaction between mutation and drift?
That brings us to the idea of mutation-drift balance. Remember that drift tends to make populations more genetically uniform,
Right, less diversity.
where everyone has the same version of a gene. Mutation, on the other hand, introduces new versions, increasing genetic diversity.
More variety.
Our source describes this as a kind of tug of war. Mutation pressure pushes towards more diversity, while drift pulls towards less. The level of genetic variation we see in a population can be a result of this ongoing balance, a tug of war. Yeah, between the creation of new diversity through mutation and the random loss of that diversity through drift, this constant tug of war highlights that the genetic makeup of small populations can be surprisingly volatile, driven by chance as much as need.
So we've got these big forces in play. But let's zoom in on mutation itself. You mentioned earlier that not all mutations are equally important in evolution. What makes a mutation really matter?
That's a key insight. Our lecture material emphasizes that the evolutionary impact of a mutation hinges on a few things: where it happens in the genome, what kind of change it is, and ultimately what effect it has, especially on the structure and function of proteins or other important RNA molecules.
Okay, location seems important. I guess a change in a working gene is more likely to have an effect than a change in a stretch of DNA that doesn't seem to do anything?
Generally, yes, but it's more complex than that. First, we need a clear idea of what a gene actually is. It's a segment of DNA that holds the instructions for building a protein or a functional RNA molecule.
The instructions.
Right. But it also includes any DNA sequence that's essential for a specific job, even if it's not directly translated into a protein. These are the working parts of our genome. Now, in more complex organisms like us eukaryotes,
Like us.
genes often have coding sections called exons and non-coding sections called introns. These introns are like interruptions in the instructions.
Introns get cut out, right?
Exactly. And the process of starting to read a gene transcription in eukaryotes is also quite intricate, involving various helper molecules that bind to the DNA and tell the machinery where to start copying.
And I remember seeing diagrams in the source showing the structure of genes in complex organisms and simpler ones, prokaryotes. They look quite different, right?
They do. Eukaryotic genes are often interrupted by these introns that need to be removed before the instructions can be used. They also have regulatory regions before the gene itself that act like control switches.
On-off switches.
Kind of. Prokaryotic genes, on the other hand, often come in groups called operons, where multiple genes are read together from a single starting point, and they generally don't have introns, much simpler structure in many ways.
And those introns in eukaryotes, they get cut out before the RNA goes on to make a protein. That's splicing, right?
Exactly. Our source had diagrams showing this. It involves these little molecular machines called spliceosomes that assemble on the RNA, form a loop, and then precisely snip out the introns and stick the exons together. It's a very precise process.
This is getting into the molecular nitty-gritty, but it makes sense that a change in one of these important regions, the coding part, the part that gets cut out, or the control switches, could have different consequences.
Absolutely. And the location story doesn't end there. We also need to think about epigenetics and RNA editing.
Epigenetics, that's about changes in how genes are used without actually changing the DNA sequence itself, right? Like tags on the DNA.
Precisely. Think of DNA mutations as long-term heritable changes that drive long-term evolution. Epigenetic changes, on the other hand, can allow for quick adjustments to the environment, but are usually not passed down to the next generation in the same way.
Faster acclimatization, maybe?
Possibly. However, there's an interesting idea that epigenetic changes might sometimes reveal hidden genetic variations, providing pre-adapted traits that selection can then act on over longer timescales.
Interesting.
Another important location-related concept is allele-specific imprinting, where only the copy of a gene inherited from either the mother or the father is active. This affects a surprisingly large number of genes in humans, around 4,000 or so.
Wow.
Then we have RNA editing, which is like a post-transcriptional modification. It happens after the DNA has been copied into RNA.
After copying.
Right. It's a widespread phenomenon where the RNA sequence itself is altered, either by adding or removing nucleotides or by chemically modifying existing ones, like changing a C to a U or an A to an I.
So the message copied from the DNA can actually be tweaked at the RNA level before it makes a protein.
That's right. And as our source hinted with those interesting article titles, RNA editing, a driving force for adaptive evolution, and non-coding RNA. What is functional and what is junk? These are really hot topics in research right now.
So mutations can not only change the underlying DNA, but also affect how that DNA is interpreted and processed into the working molecules of the cell. What about the effects of mutations that happen in the parts of the RNA that don't directly code for protein, the non-coding bits?
Well, as we've touched on, mutations in the transcribed region can directly change the protein sequence if they're in an exon, but they can also indirectly affect how that RNA is processed.
How so?
For example, a mutation might alter the signals that tell the splicing machinery where to cut and paste, messing up the introns and exons, or it could affect how the RNA is edited, where it goes in the cell, how long it lasts, or how efficiently it's translated into protein.
So lots of indirect effects.
Exactly. This can have a big impact on how much functional protein is ultimately produced. It highlights that genetic variability isn't just about differences in the protein's building blocks, but also about differences in when, where, and how much of a protein is made.
Regulation versus function.
Precisely. This brings us to a key question. Is adaptation and the emergence of new species driven more by changes in what a gene does, or by changes in how that gene is regulated, its on-off switches and dimmer controls?
Good question. And you mentioned ADAR earlier in the context of RNA editing. What's that all about?
ADAR is an enzyme that specifically changes the RNA molecule after it's been made from DNA. It converts a specific RNA building block, adenosine A, into another called inosine A.
A to I editing.
Right. What's really interesting is that the locations where ADAR acts on the RNA are determined by the underlying DNA sequence. So while the editing itself is a post-transcriptional event, the potential for it is encoded in our genes.
So the DNA sets up the editing sites.
Yes. This means that the characteristics we see, our traits, our phenotype, can be influenced indirectly through these RNA modifications. And the amount of RNA editing can even change depending on the organism or at different stages of development.
And the diagram in the source, figure 1, showed how this A to I editing can have different consequences depending on where it happens in the RNA molecule, right?
Exactly. If it happens in the part of the RNA that codes for a protein, it can change the protein's building blocks, potentially altering its function.
Changes the protein.
If it happens in a precursor to a microRNA, a small RNA that regulates other genes, it can affect how that microRNA is processed or which genes it targets.
Affects regulation.
And if it happens in the non-coding regions of the RNA, like introns or UTRs, it can influence the RNA's stability, its transport within the cell, or even how it gets spliced. It can even change the signals that tell the splicing machinery where to cut.
So RNA editing seems like a way to create even more diversity and fine-tune gene expression without making permanent changes to the underlying DNA. What's the evolutionary advantage of that?
Well, our source points out that RNA editing can be a rapid response to environmental changes, maybe short-term acclimatization.
Quick fixes?
Kind of. But it also has the potential to contribute to longer-term heritable adaptation. Most new DNA mutations are either neutral and lost by chance, or they're harmful and get eliminated by selection. RNA editing offers some benefits.
Like what?
It can create new variations at a lower evolutionary cost because the original unedited RNA can still be produced from the same gene.
You get both versions.
Potentially, yes. This might mean there's less pressure to get rid of an edited version, allowing it to stick around and be tested by selection. There's also this idea called continuous probing, or COP.
Continuous probing.
Yeah, the idea is that the editing machinery is constantly scanning RNAs, potentially generating a wider range of molecular options, sort of exploring possibilities. The diagram in our source visually suggests that RNA editing in both coding and non-coding regions can lead to a greater variety of traits, potentially leading to faster adaptation and increased biological complexity.
More phenotypes, maybe faster adaptation. Okay, this has been all about mutations in genes that are transcribed into RNA. But what about the vast stretches of DNA that don't seem to code for anything? Can changes there have an effect?
Absolutely. Our source emphasizes that even these untranscribed DNA sequences can have crucial roles. Think about the places where DNA replication starts and stops the origins and termini. Mutations there could mess with how and when our DNA is copied.
Right, fundamental processes.
Then there are the sites where the machinery of genetic recombination works during the formation of sperm and eggs. Changes here could affect how our chromosomes are shuffled. The ends of our chromosomes have protective caps called telomeres, and mutations can affect their stability.
Telomeres, right.
There are also regions where chromosomes attach during cell division, sites that bind proteins, and regions that influence the overall structure of chromosomes. Mutations in any of these seemingly non-coding regions can have significant, though often indirect, effects on the genome and how it functions.
That really highlights that non-coding doesn't automatically mean non-functional. What about pseudogenes? They sound like the ghosts of old genes.
That's a good way to put it. Pseudogenes are DNA sequences that look a lot like functional genes but have accumulated mutations that prevent them from being properly expressed or making a working product. They're like relics of genes that once worked.
Genomic fossils.
Pretty much. For the most part, they're not actively used. However, there are some interesting cases where pseudogenes are transcribed into RNA, although usually not translated into protein, and in a very few instances, they might even produce a protein fragment.
So mostly dead, but sometimes.
Yeah,
Flicker of life.
something like that. There are different ways they arise too, like gene duplication followed by inactivation, or when messenger RNA gets copied back into DNA and reinserted.
Okay, now let's get back to mutations in the protein coding regions themselves and their direct impact.
Right. In organisms like us, where we have two copies of most genes deployed organisms, a mutation in one copy will immediately affect the products of that specific copy, and generally half of the total output of that gene.
Half the protein might be different.
Potentially, yes. There are exceptions, like those imprinted genes where only one copy is active anyway, or mutations in parts of a gene that are only used in certain versions of the RNA due to alternative splicing.
Okay.
However, for many genes, there's a strong constraint. Changes to the critical building blocks of protein, the essential amino acids, are often harmful. This limits the amount of variation we see at those key positions.
Selection weeds them out.
Exactly. Purifying selection. Our source makes an important point here. If evolution relied solely on these direct mutations in protein coding sequences, the rate at which organisms could adapt might be quite slow due to these limitations.
That really underscores the importance of those other mechanisms we discussed, like changes in gene regulation and RNA editing, in driving evolutionary change. Now, mutations come in different shapes and sizes, right? We hear about tiny changes and bigger ones.
Yes, mutations can be categorized by the amount of DNA they affect. Point mutations involve a single DNA letter, a single base pair. Segmental mutations involve larger stretches, could be hundreds or thousands of base pairs.
Point versus segmental, okay.
They can also be classified by the type of change. Our source lists substitution, where one letter is swapped for another.
Swap.
Recombination, where DNA segments are exchanged usually between chromosomes.
Yes.
Deletion, where a piece is lost.
Loss.
Insertion, where a piece is added.
Added.
Inversion, where a segment is flipped around.
Flipped. Got it.
Yeah.
Let's zoom in on substitution mutations in the coding parts of genes for a moment. We often hear terms like silent, missense, and nonsense mutations. What do those mean?
Right. These terms describe what happens when a single DNA letter is changed within a protein coding gene, specifically looking at its effect on the resulting protein sequence. A synonymous mutation, also called a silent mutation, is a change in the DNA sequence that doesn't actually alter the amino acid sequence of the resulting protein.
Same protein, different DNA.
Exactly. This is possible because the genetic code has redundancy. Multiple three-letter codons can code for the same amino acid.
Okay.
A non-synonymous mutation, on the other hand, does lead to a change in the amino acid sequence. A missense mutation is a type of non-synonymous mutation where one amino acid is replaced by a different one.
Different amino acid.
And a nonsense mutation is where the DNA change creates a premature stop signal, a stop codon in the genetic code. This usually leads to a shortened and often non-functional protein.
That's a short.
Yeah. Our source also mentions the idea of a pre-termination codon, a codon that can be easily turned into a stop codon with just one single base change.
And the likelihood of these different types of mutations happening isn't equal, is it? Depends on the position in the codon.
That's correct. Our source provides some interesting data. Changes at the third position of a codon are often silent, maybe around 70% of the time, because of that redundancy in the code.
Third position is often redundant.
Right. Whereas changes at the second position are almost always non-synonymous. They always change the amino acid. And changes at the first position are also usually non-synonymous, like 96% of the time.
So second and first positions really matter for the protein sequence.
Definitely. These mutations mainly happen because of errors during DNA replication. Sometimes the DNA letters can temporarily shift into slightly different chemical forms called tautomers, leading to incorrect pairings when the new strand is being made.
Mispairing during copying.
Exactly. There are two main types of these substitutions. Transitions, which are swaps between similar types of DNA letters, like one purine for another purine, or one pyrimidine for another pyrimidine.
Similar swap.
And transversions, which are swaps between a purine and a pyrimidine.
Different type swap.
Right. These happen at different frequencies.
These are single letter changes.
Yeah.
But when we compare the DNA of different species, the patterns of change can be quite intricate, right? Not just single swaps.
Absolutely. Our source uses the beta-globin gene as an example to illustrate some of these complexities when comparing sequences from different organisms. We can see parallel substitutions,
Like parallel.
where the same change happens independently in different evolutionary lines. There are single substitutions, just one change along a branch of the evolutionary tree. Sequential substitutions involve multiple changes at the same spot in a single lineage.
One spot changing multiple times.
Yeah. Coincidental substitutions are different changes in different lineages that end up with the same final DNA letter by chance. Convergent substitutions are where different changes in different lineages lead to the same amino acid if it's a protein coding change.
Ending up the same place via different routes.
Exactly. And then there are back substitutions where a site changes and then changes back to its original state, which can sometimes hide the total number of changes that have actually occurred.
It's like trying to piece together a history book where some of the pages have been rewritten multiple times and sometimes erased. Now, you mentioned that mutations don't happen randomly across the entire genome. There are these hotspots. What makes some regions more prone to mutation?
Exactly. Mutation hotspots are regions of the genome that have significantly higher rates of mutation than the average. Our source gives several examples. Sequences with the letters C followed by G, CpG dinucleotides, are often hotspots in eukaryotes like us. The C can be chemically modified, methylated, and this modified C is prone to deamination, turning it into a T. So CG often becomes TG over time.
Oh.
In simpler organisms, like bacteria, sequences with two T's in a row, TT, can be hotspots. Palindronic sequences, which read the same forwards and backwards on opposite strands, can also be prone to mutation in bacteria.
Palindrome.
And in eukaryotes, short, repeated DNA sequences like CACA or just stretches of AS are often hotspots for additions and deletions in DELs due to a kind of slippage during DNA replication. The machinery sort of stutters over the repeats.
Lip strand mispairing.
Right. Our source even referenced a research paper specifically on hotspots of human mutation, showing how important this non-randomness is.
Knowing about these hotspots is probably really important when trying to understand evolutionary changes in DNA. Now, how do we actually analyze all these substitutions to understand how different organisms are related? I remember something about Ds and DN.
Yes, DS versus DN. Looking at the patterns of synonymous DS and non-synonymous substitutions in the DNA that codes for proteins is a really powerful way to study evolutionary relationships and selection pressures.
Synonymous doesn't change the protein, non-synonymous does.
Correct. Because synonymous changes don't usually affect the protein's function, they're often under less pressure from natural selection and tend to accumulate more readily over time. We often consider them neutral or nearly neutral.
So they build up more easily.
Generally, yes. This makes the DNA sequence, with both types of changes, very useful for building evolutionary trees, especially for comparing more closely related species where protein sequences might be very similar.
Right.
By comparing the rates of these two types of changes, Ds and Dn, we can actually get clues about what kind of selection is acting on a gene. If the rate of synonymous changes is much higher than the rate of non-synonymous changes, so DS-Dn.
Lots of silent changes. Few protein changes.
Right. It suggests that the gene is under purifying selection, meaning that most changes to the amino acid sequence are harmful and are being eliminated by selection. The protein sequence is being conserved.
Okay, purifying selection.
If, on the other hand, the rate of non-synonymous changes is higher, D and DS, it suggests positive selection, where changes to the protein sequence are actually beneficial and are being favored and spread by evolution. The protein is adapting.
Positive selection, driving change.
And if the rates are roughly equal, DS-DN, it might mean that the gene is evolving neutrally, with most changes having little to no impact on fitness, and their fate is determined more by genetic drift.
Neutrality.
Yeah. This idea is central to the neutral molecular evolution theory, proposed by Motu Kimura back in '68, which posits that many, perhaps most, of the genetic differences we see between species are due to the random accumulation of neutral or nearly neutral mutations through genetic drift.
Drift is key again.
Exactly. Our source included an example showing aligned protein and DNA sequences from different species, highlighting the sites that have changed and how we can compare these rates visually.
So, looking at these DS-DN rates can tell us if a gene is being conserved, actively changing to something new, or just drifting randomly.
Oh, yeah.
What else do we need to keep in mind when we're analyzing these substitutions?
Our source mentions a few other important things. One is that it's often helpful to compare more than just two sequences. If we have a third, more distantly related sequence, an outgroup, basically.
An outgroup.
It can help us figure out the direction of changes and identify potential back substitutions, which we wouldn't see by just comparing two sequences.
Helps root the changes.
Exactly. We can also look at non-coding regions of the DNA, like the UTRs, untranslated regions, or even introns sometimes. These regions might be under less strict functional constraints and could show more differences, useful for different evolutionary timescales.
Non-coding regions, too.
And pseudogenes, those fossil genes, and other inactive DNA elements can also act like molecular fossils, accumulating changes at a relatively steady rate, potentially reflecting the underlying mutation rate more closely.
More clues there.
And finally, it's often useful to look at the rates of transitions, AG, CT, separately from transversions, pure iron pyrimidine, as these types of changes often happen at different frequencies. Some models account for this.
Okay. Now, once we've looked at these substitutions, how do we actually calculate how far apart two sequences or two species are in evolutionary terms? How do we get a distance measure?
Right, the evolutionary distance. The simplest way is just to line up the two sequences and count the number of positions where they differ, then divide by the total length. That's the Hamming distance or p distance.
Just count the differences.
Yeah, the raw proportion of difference. However, this observed difference might underestimate the true evolutionary distance because it doesn't account for the possibility that a particular DNA letter might have changed multiple times over evolutionary history, but we only see the final state. Like a site changing from A to G and then back to A, we wouldn't see any change.
Hidden mutations.
Exactly. To correct for these hidden or multiple mutations, we use evolutionary distance models. One of the earliest and simplest is the Jukes and Cantor model, often called JC69.
Jukes and Cantor.
It uses a mathematical formula involving a logarithm to estimate the true number of substitutions per site based on the observed proportion of differences. It essentially assumes that if you see more differences, it's increasingly likely that multiple hits have occurred at some sites.
Tries to correct for the hidden stuff.
Right. But its key assumption is that all types of DNA letter changes, A to T, A to C, A to G, etc., are equally likely.
Which isn't always true, right? Transitions versus transversions.
Exactly. We know transitions often happen more frequently than transversions. So the JC69 model is generally okay for sequences that aren't too different, maybe less than 70% difference, but it can be inaccurate for more distant relationships.
So for sequences that are more distantly related, we need more complex models.
Exactly. Kimura's K82 parameter model was a big step forward. It specifically takes into account that transitions and transversions often occur at different rates. It basically gives more weight to the rarer transversion changes when calculating distance.
It accounts for the two rates.
Right. And then Tamura's T92 model goes even further by also considering the overall composition of the DNA, specifically the proportion of Gs and Cs versus As and Ts, the GC content, which can vary between different genomes and influence mutation patterns.
Yeah, it's GC content.
Yeah. Our source had diagrams that illustrated how these different models account for different types of changes in base frequencies. So the key idea is that these models help us get a more accurate estimate of evolutionary distance by trying to account for the complexities of how DNA actually mutates over time.
And these models seem to assume that the rate of change is the same across all positions in a DNA sequence, right? But I'd imagine, like you said, some parts of a protein or RNA might be more important and therefore change more slowly than others.
That's a very insightful point and a crucial one. In reality, there's often significant variation in the rate of change across different sites in a sequence. Rate heterogeneity.
Heterogeneity.
Some positions might be highly conserved because they're crucial for function, maybe an active site in an enzyme or a structural part of an RNA. Changes there are strongly selected against. Other positions might be less important and can change more freely.
Variable speeds along the sequence.
Exactly. We see this, for example, in protein coding genes. The third codon position often has a higher rate of change because many mutations there are synonymous. Within a protein, the core functional domains might be very stable, while surface loops could change more readily. Similarly, in RNA molecules, regions that form important structures, like stems in a hairpin, might be conserved, while loops could vary more.
Makes sense. How do we account for that?
To deal with this rate heterogeneity, scientists use models that incorporate rate variation. The most common approach is the gamma model.
The Gamma model.
This model assumes that the rates of change across different sites follow a statistical distribution, specifically the gamma distribution. The shape of this distribution is determined by a parameter called alpha.
Alpha.
A large alpha value means the distribution is narrow, indicating that most sites are changing at roughly the same, relatively slow average rate. A small alpha value means the distribution is broad and L-shaped, indicating there's a lot of variation. Many sites are evolving very slowly, highly conserved, while a few sites are evolving very quickly.
So alpha tells you how variable the rates are.
Precisely. Genes with a small alpha often have a low overall average rate of change because so many of their sites are under strong constraint, even if a few sites change rapidly. Using a gamma correction helps get more accurate distances, especially for longer evolutionary times.
So using these different, more sophisticated models allows us to get a more refined picture of evolutionary distance by acknowledging that not all DNA changes at the same pace, either in terms of type or location. Now let's talk about the molecular clock. It sounds like something that would allow us to put dates on evolutionary events.
That's precisely the idea behind it. The molecular clock hypothesis, which really took off in the 1960s with people like Zucker-Kandel and Pauling, suggests that for any given protein or gene, the rate at which its sequence changes through evolution is roughly constant across different evolutionary lineages.
The steady tick-tock of changes.
Kind of. The basic idea is that if mutations accumulate at a relatively steady pace, we can use the amount of molecular difference between two species corrected using those models we just discussed to estimate how long ago they shared a common ancestor.
Using molecular distance to tell time.
Exactly. Researchers like Richard Dickerson did some seminal work on this. He plotted the time of divergence between species, estimated from the fossil record, against the amount of protein sequence difference corrected for multiple hits, for proteins like cytochrome C, hemoglobin, and fibrinopeptides.
What did he find?
He had found a fairly straight-line relationship for each protein, suggesting a somewhat constant rate of change over millions of years for that specific protein. However, he also noticed that different proteins evolved at different average rates. Cytochrome C changed very slowly, while fibrinopeptides changed much faster.
Different clocks for different proteins.
Right. He concluded that these different rates likely reflected how critical each protein's function is and how much change it could tolerate. Proteins with very important constrained jobs tend to evolve more slowly because most changes would be harmful and selected against.
Functional constraints set the clock speed.
That's the idea. This opened up the exciting possibility of using these molecular clocks to estimate divergence times for groups where the fossil record is sparse or missing. It's important to remember, though, that this clock reflects the rate of substitution. The changes that actually become fixed in a population, not necessarily the underlying mutation rate, which might be higher, but includes many changes that are quickly eliminated by natural selection.
So it's not a perfect timekeeper, maybe more like a rough chronometer, but it's a valuable tool for getting a sense of the timeline of evolution. Are there situations where this idea of a constant rate doesn't hold up, so the clock always tick at the same speed?
No, not always. And that leads to the concept of a local clock. Sometimes the rate of molecular evolution might be relatively constant within a specific group of closely related organisms, but different from the rate in other lineages.
Constant just for that group.
Right. Our source gave an example comparing the rates of synonymous DS and non-synonymous DN changes in mice and rats, using hamsters as a more distant relative, an outgroup. They looked at the changes on the branch leading to mouse since the split from hamster, and the changes on the branch leading to rat since the split from hamster.
And what did they find?
The fact that the DIDS rates were similar between the mouse lineage and the rat lineage, and the DN rates were also similar between the two lineages, supported the idea of a local clock operating fairly consistently in the mouse and rat lineages since they diverged from the hamster lineage.
So the clock was ticking similarly for mice and rats in this case.
Yes, for both synonymous and non-synonymous changes, which strengthens the argument.
And what about broader exceptions to the molecular clock idea? Are there times when the rate of molecular evolution really isn't constant across major groups or over long periods?
Absolutely. The rate of molecular evolution isn't always the same across all organisms, all genes, or even all parts of a single gene. It can vary quite a bit.
What causes the variation?
For example, viruses tend to evolve much, much faster than many other organisms due to their rapid replication rates and sometimes less accurate copying mechanisms. The main reason for these variations generally speaking, is natural selection.
Selection again?
Yes, genes that are under strong purifying selection to maintain their function will evolve slowly. Genes or regions under relaxed constraint, or especially under positive selection to adapt to new conditions, can change much more rapidly.
Positive selection speeds it up.
It certainly can. Organisms with shorter generation times or faster metabolisms are also sometimes observed to have faster molecular clocks, though the reasons for this are still debated, and the patterns can be complex across different life forms. For reliable molecular clock analysis, it's usually best to choose genes whose function is relatively consistent and conserved across the groups you're studying.
Choose your gene carefully.
Definitely. We also need to be aware of the effects of positive selection, which can cause bursts of rapid change, and negative purifying selection, which slows down evolution by removing harmful mutations. These can both make the clock seem uneven.
So there are a lot of factors that can influence how quickly or slowly the molecular clock ticks in different lineages. What are some of the specific biological things that might cause these differences in rate?
Our source delves into factors related to DNA replication itself and those that are kind of independent of it.
Okay, replication dependent.
Yeah, these include things like generation time and the efficiency of DNA repair systems. The generation time idea suggests that organisms with shorter reproductive cycles, like rodents compared to humans.
Mice versus people.
Right. They might go through many more rounds of DNA replication in their germline over a given period, say 1,000,000 years. If mutations primarily happen during replication, then more replication cycles could lead to more accumulated mutations per unit of time.
More copying, more chances for mistakes.
That's the logic. There's some evidence for this, particularly in the nuclear DNA of mammals, though it gets complicated when comparing nuclear and mitochondrial DNA rates. Also, the efficiency with which an organism can repair damaged DNA can play a role. Less efficient repair systems might lead to a higher mutation rate persisting. There was Britton's hypothesis about this.
Better repair, slower clock.
Potentially, yes. Then there are replication-independent factors.
Like what?
Things like basal metabolic rate and maybe body size, although that's often correlated with metabolism and generation time. The metabolic rate hypothesis suggests that organisms with higher metabolic rates might produce more damaging byproducts, like oxygen radicals, which can damage DNA even when the cell isn't dividing.
More metabolic activity, more DNA damage.
That's the idea. This could lead to higher mutation rates independent of replication. There's some evidence linking metabolic rate to evolutionary rate, for instance, comparing cold-blooded versus warm-blooded animals. But the patterns aren't always clear-cut across all groups of life. It's an area of ongoing research.
Wow, this has been an incredibly deep dive. We've covered so much ground. From the fundamental idea of using molecules' DNA, proteins to trace evolutionary history,
Molecular phylogeny, yeah.
to the complex forces that shape our genomes, drift, selection, mutation, and their balance.
That dynamic interplay.
Then we got into the different kinds of mutations where they have a coding, non-coding, introns, epigenetics, RNA editing.
All those layers of complexity.
How we analyze these changes, comparing synonymous and non-synonymous rates, using models like Jukes-Cantor, Kimura, Tamara, accounting for rate heterogeneity with gamma.
Getting those distance estimates right.
And finally, the fascinating, though nuanced concept of the molecular clock, its potential for dating evolution, but also its exceptions and the factors like selection, generation time, and metabolism that influence its ticking.
Absolutely. And hopefully you, our listener, now have a much richer understanding of how we can read the history of life written in the very fabric of our being, right down at the molecular level.
Yeah.
This knowledge allows us to see the interconnectedness of all living things at the most fundamental level and to appreciate the epic journey of evolution that led to everything we see today.
It really is an amazing story hidden in our genes.
Yeah.
And on that thought, here's something to ponder. Consider how this seemingly random process of genetic drift, which we talked about as this force driven by chance.
The random fluctuation.
Right. How can that process have such profound and lasting effects on the evolution of life, sometimes even overshadowing the more directed power of natural selection? What could this imply about the future evolutionary paths of small, isolated populations, especially in our rapidly changing world today?
That's a really interesting question to think about, the power of chance in evolution.
Exactly. Something to mull over. Thanks for taking this deep dive with us. We encourage you to keep exploring these fascinating connections on your own.